Have you ever stopped to think about how your body manages to, well, power itself? Not just when you're enjoying a big meal, but, even through those long stretches between meals. Or when you're really pushing yourself. It's kind of a masterful act of biological juggling, isn't it? Today, we're doing a deep dive into metabolism. We're drawing from some foundational stuff like boron and Bullpeep's medical physiology. Our mission really is to make these super complex ideas clear, engaging, and frankly, accessible for you. Whether you're a college student, knee-deep in med school, or just really curious about how your body ticks.
Yeah, and what's really fascinating, I think, is how we can start with the big picture, right?
Yeah.
And then carefully peel back the layers, sort of step by step, to understand the nuts and bolts of how these systems actually work. And we'll try to connect everything to real-world relevance, too, so you can see how it all ties into, diagnostics, maybe pathology, treatment, hopefully giving you those aha moments.
Exactly. Those moments where it just clicks. So we're going to cover the basics, what metabolism even is. Then we'll unpack the different energy forms your body uses, touch on the universal laws governing energy. Then we'll get into the core pathways for energy conversion and use. And finally, how the body adapts, both to eating and, maybe more dramatically, how it survives during fasting.
The grand orchestra of metabolism setting the stage.
Okay, so let's kick things off. What is metabolism, basically?
Well, it's really the sum total of all the chemical reactions happening in your body that involve energy. Think of it like a giant orchestra. You've got some processes building things up. We call those anabolic processes, like building muscle protein. And then you have processes breaking things down, catabolic processes, like breaking down glucose for energy. It's this constant dynamic balance. Right. And absolutely all the energy you get from food eventually shows up as either heat or work your body does, moving, breathing, even thinking or growth, which is basically stored energy. And your body needs energy constantly, even just sitting there. Take a healthy adult, for example. Just for their basic functions, they need a fair amount of energy each day. That's the resting metabolic rate, or RMR. Now, for clinical purposes, there's a stricter measure, the basal metabolic rate, BMR. It's measured under really specific standardized conditions. Quiet room, after a fast, completely rested. It's usually a bit lower than RMR, and it tends to decrease as we get older.
Yeah, and what always gets me is the challenge the body faces. You eat, what, maybe 3 times a day? But you're using energy continuously, your brain never really sleeps, your heart keeps pumping, so the body has to be incredibly smart about storing energy and then doling it out carefully when needed.
Absolutely. And that whole process, that dance, is orchestrated by this complex interplay. You've got the nutrients coming in, hormones acting as signals, and constant chatter, fuel exchange between organs. Insulin is really the star player here. It's the main hormone directing traffic in both the fed state after you eat and the fasted state, but others jump in too. Glucagon, catecholamines, think adrenaline, cortisol, growth hormone. They're important for more acute energy needs. And the major organs, well, the liver is like the central fuel depot. It's the main producer of glucose, usually the only one, actually. The brain is a huge consumer, almost totally dependent on glucose, especially when you're fed or just starting a fast. And then your muscle and adipose tissue, that's your fat, they're the big storage sites. They listen to insulin and store energy as glycogen and fat.
That makes a lot of sense. So where does this energy actually originate? Let's unpack the specific fuel types the body uses.
The body's fuel pantry, carbohydrates, proteins, and lipids.
Okay. Fundamentally, pretty much all the energy we use comes from breaking carbon bonds. Bonds originally created by plants using sunlight photosynthesis. We get this energy from 3 main types of molecules in our food: carbohydrates, proteins, and lipids or fats. Each has its own building blocks. For carbs, it's simple sugars like glucose, fructose, galactose. For proteins, it's amino acids. And for lipids, fatty acids.
Right. Let's start with carbohydrates. They give you a decent energy kick, about 4 kilocalories per gram. The main way your body stores carbs is as glycogen. You can picture it like a big branching tree with glucose molecules as the leaves. Alpha 114 links in the straight bits, alpha 16 at the branches. It's stored pretty much everywhere, but the big depots are your liver and muscles. The liver holds a smaller but really vital amount, maybe 75, 100 grams. It's crucial for keeping your blood sugar steady for the brain. Your muscles, though, they hold the largest total amount of glycogen, maybe 300, 400 grams, just because you have so much muscle. But that's mainly for the muscle's own use. Okay, but here's something interesting about glycogen. It's not the most efficient way to store energy in terms of space, because for every gram of glycogen, you store about 1 or 2 grams of water right along with it, which makes it bulkier than fat.
Exactly. It makes it less energy dense per hydrated gram. But that water is important for the cell too. And clinically, this difference between liver and muscle glycogen is huge. The liver has a special enzyme, glucose 6-phosphatase or G6-PASE. Muscles don't. So the liver can break down glycogen and release free glucose straight into the blood, vital if your blood sugar drops. Muscle glycogen, it breaks down to glucose 6-phosphate, which gets used right there in the muscle cell for energy, like during a sprint. It can't leave the muscle to boost blood sugar.
Got it. Liver glycogen is for sharing. Muscle glycogen is local fuel.
Okay, next up, proteins. These are long chains of amino acids. Some are essential, meaning you absolutely have to get them from your diet because your body can't make them. Energy-wise, proteins are similar to carbs, around 4 kilocalories per gram. Now, you have a lot of protein in your body, maybe close to 10 kilograms in a 70-kilogram person, but only about half of that is really accessible as fuel in a pinch. The main point here is proteins are not your go-to energy reserve. Their primary jobs are structural, think skin, collagen, and functional enzymes, muscle filaments, hormones. Normally, protein breakdown provides less than 5% of your resting energy, but during starvation, that can jump up maybe to 15% at a last resort. Any extra protein you eat gets burned or turned into glycogen or fat.
And now for the really interesting one, lipids, fats. These are by far the most concentrated energy source. We're talking over 9 kilocalories per gram, more than double carbs or protein.
Right. And the reason they're so efficient is they're stored without much water, almost pure fuel, packed tightly. This density is absolutely crucial. Think about trying to move around carrying weeks' worth of energy stored like bulky glycogen. Fat storage makes us mobile, and it's key for surviving famine.
So where is it stored?
Mostly in your adipose tissue, under the skin and around organs, also some in muscle. A typical 70-kilo person with 20% body fat is carrying around 14 kilograms of fat. That's potentially over 130,000 kilocalories, enough to fuel resting metabolism for, theoretically, nearly 9 weeks.
Wow, so fat really is the major fuel tank for the long haul.
The universal laws of energy and the body's currency, ATP. Okay, to really understand how the body manages all this fuel, we need a quick nod to the laws of thermodynamics. The first law is simple. Energy isn't created or destroyed. For your body, it means energy in food must equal energy out, work, heat, synthesis, plus any energy stored. So positive energy balance, eating more than you burn, means storage, usually as fat. Negative balance means using up stores, losing weight. We also sometimes talk about nitrogen balance for protein stores. And the second law, that tells us about efficiency. It basically says that whenever energy changes form, some usable energy is always lost, mostly as heat. Chemical reactions are never 100% efficient. So when your body does something like convert glucose to glycogen, some energy just unavoidably dissipates as heat. It's a fundamental rule.
Exactly. And that brings us neatly to ATP, adenosine triphosphate. This is the body's main energy currency, like the dollar of the cell. Structurally, it's simple. Adenine, a ribose sugar, and 3 phosphate groups. The bonds connecting the last 2 phosphates are often called high-energy bonds. When one of those bonds breaks, it releases a packet of energy the cell can use immediately. What makes ATP so useful is its energy level. It's sort of in the middle. It can accept energy from breaking down high-energy molecules like fuel sources and then donate that energy to power other reactions like muscle contraction or pumping ions.
Like charging and discharging a battery.
Precisely. It fuels things like myosin heads, grabbing actin in muscle, or the pumps that move calcium back into storage to relax the muscle, and it shows the body's efficiency. If you just burn glucose in a lab, all its energy becomes heat. In the body, a good chunk, maybe 40% or so, is captured as ATP, even though some is still lost as heat due to that second law. Still pretty impressive.
The core metabolic pathways, glycolysis and gluconeogenesis.
All right, let's zoom in now on some really central pathways. Specifically, how the body deals with glucose, a 6-carbon sugar, and pyruvate, a 3-carbon molecule.
First, glycolysis. This is the breakdown of one glucose molecule into 2 pyruvates. It's fundamental. And it can happen with oxygen aerobic or without anaerobic. Even anaerobically, it nets you a quick 2 ATP molecules per glucose. With oxygen, the pyruvate goes on to yield much more ATP later. Clinically, this is vital. Cells like red blood cells, which lack mitochondria or fast-twitch muscle fibers during intense bursts, rely solely on this fast anaerobic glycolysis.
And its speed is controlled.
Tightly regulated. Key enzymes get slowed down by products like ATP when energy is high and sped up when energy like AMP is low. Oxygen availability also plays a role indirectly. And here's a key clinical point. In anaerobic conditions, that pyruvate gets converted to lactate, If this happens too much, you get lactic acidosis. That acid buildup lowers pH, makes muscles fatigue, causes cramps, and can even inhibit enzymes. It really underscores why sustained activity needs oxygen.
Okay, so glycolysis breaks glucose down. What about building it up? That's gluconeogenesis, making new glucose from things that aren't sugars. And this is where it gets really interesting because it's absolutely essential for life. Why?
Your brain and other tissues like red blood cells, renal medulla, they normally depend on glucose. The adult brain alone chews through about 120 grams of glucose every single day,
So where does this happen?
primarily the liver. The kidney cortex helps out too,
Yeah.
especially during prolonged fasting, maybe contributing up to 40%.
Now, critically, gluconeogenesis is not just glycolysis in reverse. It actually costs energy, ATP, and GTP to make glucose this way.
It cleverly bypasses the 3 irreversible steps of glycolysis using 4 unique enzymes. Think pyruvate carboxylase, PPCK, fructose 1,4,6-bisphosphatase, and that liver-specific G6-base we mentioned.
And the body keeps these pathways separate.
It does, partly through compartments. Some enzymes are in the mitochondria, some in the cytosol, G6-base is in the ER. This minimizes wasteful, futile cycling, the building blocks. Lactate, pyruvate, amino acids, especially alanine and glutamine and glycerol, from fat breakdown. But remember, fatty acids themselves generally can't be turned into glucose in humans. Their breakdown product, acetyl-CoA, can't make net pyruvate or oxalacetate, which leads to reciprocal regulation. This is crucial. Since both glycolysis, breakdown, and gluconeogenesis build up release energy overall, running both full tilt at the same time would just burn ATP for nothing, a true futile cycle. So they're tightly coordinated. Short term, by molecule signaling energy status. For example, high energy signals like ATP tend to inhibit glycolysis and promote gluconeogenesis. Low energy signals like AMP do the opposite. There's also a key regulator molecule, fructose 2,6-bisphosphate, controlled by insulin and glucagon, that acts like a switch. High levels favor glucose use, low levels favor glucose production. Longer term, hormones like insulin, glucagon, epinephrine, and cortisol actually change the amount of these key enzymes the cell makes through gene expression. Insulin pushes towards glucose use and storage. Glucagon and epinephrine push towards glucose production and release. It's beautifully coordinated.
Fat synthesis and the limitations of fuel interconversion.
Okay, so the body can make glucose. What about making fat? How does the liver convert extra glucose or amino acids into fatty acids?
Right, this happens mainly when energy intake exceeds demand. Glucose goes through glycolysis to pyruvate. Pyruvate enters the mitochondrion. Now, if ATP levels are high, the usual pathway for pyruvate is slowed down. Instead, pyruvate helps make citrate. This citrate then gets shuttled out of the mitochondrion into the cytosol. Think of it like a taxi service for carbon atoms. Citrate gets back out, and an enzyme called citrate lyase splits it, releasing acetyl-CoA right there in the cytosol. And that acetyl-CoA is the starting material for building new fatty acids.
So acetyl-CoA essentially moves from mitochondria to cytosol.
Exactly. Some amino acids can also feed into this acetyl-CoA pool. And fatty acid synthesis itself happens in the cytosol. The key control step is making malonyl-CoA from acetyl-CoA. This requires ATP and an enzyme called acetyl-CoA carboxylase, or ACC, that's the rate limiter. Then, 2 carbon units from malonyl-CoA are added repeatedly to build up the fatty acid chain, say to make palmitate. Finally, these fatty acids get attached to a glycerol backbone to form tags. The liver packages these tags into VLDLs, very low-density lipoproteins, and ships them out into the blood to be stored elsewhere, mainly in adipose tissue.
It's fascinating how flexible the body is, but you mentioned limitations earlier. There's a sort of hierarchy, right?
Yes, this is a really critical point to grasp. While amino acids can become glucose or fat, and glucose can definitely become fat and some non-essential amino acids, fat generally cannot be converted back into either glucose or amino acids in humans. Why?
Yeah.
Because when you break down fatty acids, you do it 2 carbons at a time, making acetyl-CoA. In the citric acid cycle, those 2 carbons from acetyl-CoA are eventually lost as CO2. There's no net gain of carbon skeletons that could be used to build glucose.
Unlike plants, which have a way around this.
Exactly. Plants have the glyoxylate cycle. We don't. So for us, fat is basically for storage or for burning directly. It's a one-way street out of fat for glucose production.
Energy capture and metabolism. What happens after a meal?
Okay, let's switch back to the fed state. What happens right after you eat? And it's worth remembering, just processing food costs energy, right? The thermic effect of food.
Yes. Digesting and storing food raises your metabolic rate temporarily. Storing carbs or lipids costs a bit of energy, maybe 3–7% of their energy content. Storing protein is actually more costly, around 25%. And converting one fuel type to another costs even more. So, after a carbohydrate meal, the main goal is to manage blood glucose. 3 things happen. Your liver stops making glucose, your liver starts taking glucose up, and your peripheral tissues, mainly muscle, take up glucose. Insulin is the key signal, boosted by glucose itself and gut hormones called incretins. The liver acts like a buffer. Insulin rises, glucagon falls. It takes up glucose, stores it as glycogen. If there's excess glucose, it turns it into fatty acids, packages them as VLDLs. Muscle is actually the main player in clearing glucose from the blood after a meal. It stores it as glycogen or burns it. Insulin helps by moving GLUT4 transporters to the cell surface, opening the door for glucose. Adipose tissue uses a smaller amount of glucose, but it's crucial. It uses glucose to make the glycerol backbone needed to store fatty acids as tags. These fatty acids mostly come from those VLDLs the liver sent out, or chylomicrons from dietary fat.
Okay, what about after a protein meal?
Absorbed amino acids can be used for energy, but their main fate is building or repairing proteins. Liver grabs a good portion, especially those that can be turned into glucose if needed. Muscles are particularly keen on branched-chain amino acids, leucine, isoleucine, valine, using them to stimulate protein synthesis and reduce breakdown. Insulin plays a big role here too, mainly by suppressing protein degradation. Interestingly, protein alone stimulates insulin, but glucagon also rises to prevent the insulin from causing low blood sugar by keeping liver glucose output going. And finally, after a fatty meal, dietary fats, mostly tags, are broken down, absorbed, rebuilt into tags inside intestinal cells, then packaged into chylomicrons. These enter the lymph, then the blood. Insulin, stimulated by any carbs or protein in the meal, is key for handling these fats. First, it activates lipoprotein lipase, LPL, on blood vessel walls, especially near fat tissue. LPL breaks down the tags in chylomicrons and VLDLs, releasing fatty acids that fat cells can take up and restore. Second, insulin boosts glucose uptake into fat cells via GLUT4. Fat cells need this glucose to make glycerol 3-phosphate, the backbone for storing those incoming fatty acids as tags. They can't easily use free glycerol. Third, insulin strongly inhibits hormone-sensitive lipase, HSL, inside the fat cells. This prevents the breakdown of already stored fat.
So it's promoting storage and blocking release.
Exactly. In a mixed meal, insulin orchestrates everything towards net storage. Glycogen synthesis up, breakdown down, protein synthesis up, breakdown down, fat synthesis and storage up, breakdown down. It's all about capturing and conserving energy.
Energy liberation, catabolism,
Yep.
surviving during fasting and exercise.
All right, let's flip the coin. How does the body release energy when needed? During fasting or exercise, the basic idea is breaking down the stored stuff, right? Glycogen, tags.
Precisely. Breaking down those complex polymers into simpler molecules, cells can burn for ATP. Let's start with glycogenolysis, glycogen breakdown. In skeletal muscle, this is triggered by things like epinephrine, adrenaline, or even just muscle activity itself via AMP and calcium. It breaks glycogen down to glucose 1-phosphate, then glucose 6-phosphate, which is used right there in the muscle cell. In the liver, it's similar, but crucially different. Glucagon is a major trigger here, along with epinephrine. Glycogen breaks down to G1P, then G6P, but the liver has G6-pase. It snips off the phosphate, releasing free glucose into the blood.
So muscle glycogen for local sprints, liver glycogen for the whole body marathon, essentially.
That's a good way to think about it. Liver glycogenolysis maintains blood glucose, especially for the brain. Then there's lipolysis in fat cells, releasing stored fat. Hormone-sensitive lipase, HSL, is the key enzyme. It's activated by hormones like epinephrine and growth hormone, often via CAMP-P. HSL breaks down tags, releasing fatty acids, which bind to albumin in the blood, and glycerol, which heads to the liver.
Okay, now for the fatty acids themselves, how are they burned? That's beta oxidation.
Right. This happens inside the mitochondrial matrix. Remember, synthesis was in the cytosol. First, fatty acids need to get inside the mitochondria. They use the carnitine shuttle. Basically, the fatty acid gets activated, attached to carnitine, transported across the inner mitochondrial membrane, then handed back to CoA inside the matrix. Carnitine recycles back out.
A clever transport system.
Very. Once inside, beta oxidation is a cycle. It repeatedly chops off 2 carbon units as acetyl-CoA, generating energy carriers, NADH and FADH2, with each chop, until the whole fatty acid chain is consumed, a critical point. Unlike glycolysis, beta oxidation absolutely requires oxygen to ultimately yield ATP. And there's that regulatory link we hinted at. Malonyl-CoA, the product of the first committed step in fat synthesis, actually inhibits the carnitine shuttle, specifically CAT. So when you're making fat, you automatically shut down fat burning, prevents futile cycling. Now, all these breakdown pathways, glycolysis producing pyruvate, which becomes acetyl-CoA, beta oxidation producing acetyl-CoA, even amino acid breakdown contributing, they all feed into the final common pathway. That's the citric acid cycle, also called Krebs or TCA cycle, and oxidative phosphorylation. Acetyl-CoA enters the citric acid cycle in the mitochondrial matrix. Its two carbons are ultimately released as CO2, and the cycle captures energy as GTP and ADH and FADH2. The cycle is tightly regulated by how much substrate is available and by feedback from its own products.
And then oxidative phosphorylation uses those energy carriers.
Exactly. NADH and FADH2 deliver their high-energy electrons to the electron transport chain on the inner mitochondrial membrane. As electrons are passed along, protons are pumped out, creating a gradient. Then, as protons flow back in through ATP synthase, ADP is phosphorylated to make lots and lots of ATP. That's where the vast majority of ATP comes from aerobically.
Okay, but what happens if beta oxidation is running really fast, maybe faster than the citric acid cycle can handle? Or if glucose is super scarce, that leads to ketogenesis, right?
Yes. This happens typically during prolonged fasting, very low-carb diets, or uncontrolled type 1 diabetes. What these states have in common is accelerated fat breakdown producing tons of acetyl-CoA and often accelerated gluconeogenesis, which is pulling intermediates out of the citric acid cycle. The result? The liver mitochondria start converting excess acetyl-CoA into three ketone bodies: acetoacetate, beta-hydroxybutyrate, and acetone. Acetoacetate itself is acidic, contributing to metabolic acidosis if levels get too high.
Ah, the ketoacidosis link.
Precisely. The liver can also convert acetoacetate to beta-hydroxybutyrate or to acetone, which is volatile. That's the source of the fruity breath sometimes detected in diabetic ketoacidosis. But here's the crucial part. While the liver makes ketones, it lacks a key enzyme to use them. So ketones build up in the blood and are exported to other tissues: brain, heart, skeletal muscle. They can take up ketones and convert them back to acetyl-CoA to use as fuel in their own citric acid cycles. It's a fuel sharing mechanism. It's also interesting to compare fuel efficiency differently. Fats have the most energy per gram, sure, but per liter of oxygen consumed, carbs and fats are actually pretty similar.
So why prefer one over the other?
It comes down to oxygen availability and speed. Carbs give you ATP faster, especially anaerobically, making them ideal for short bursts. Think fast-twitch muscle fibers. Fats, yielding huge amounts of ATP per molecule, like 106 ATP from palmitate, are better for sustained aerobic activity where oxygen isn't limiting. Think slow-twitch muscle fibers in a marathon. We can measure fuel use with the respiratory quotient, RQ, the ratio of CO2 produced to O2 consumed. For pure carbs, it's 1.0. For pure fat, it's about 0.7. Protein is around 0.8. After an overnight fast, the whole body RQ is usually around 0.8, showing we're burning a mix of fuels. Tissues like the brain, using mostly glucose, have an RQ near 1.0.
Integrative metabolism of fasting, the body's masterful adaptations.
Okay, let's bring it all together now.
Yeah.
Fasting. How does the body adapt? You said the priorities are keeping the brain fueled and preserving protein.
Exactly. Let's start with the overnight fast as our baseline. After 12 hours or so, your brain is still demanding glucose, maybe 4 to 5 grams an hour. Your easily accessible glucose stores are pretty small, only enough for a couple hours. So the liver steps up. At this point, about half the glucose production comes from breaking down stored liver glycogen, and the other half comes from gluconeogenesis, making new glucose. We see those cycles in action. The Cori cycle recycles lactate from anaerobic tissues, like red blood cells, back to the liver to make glucose. The energy for this comes from the liver burning fat and the glucose-alanine cycle. During fasting, muscle breaks down some protein, releasing amino acids, especially alanine. Alanine goes to the liver, provides carbons for gluconeogenesis, and importantly, helps transport nitrogen safely to the liver for disposal as urea. Meanwhile, lower insulin levels allow lipolysis to ramp up. Fatty acids are released from fat stores to fuel most other tissues, sparing glucose. Glycerol released also goes to the liver for gluconeogenesis.
Okay, but what happens if the fast continues? Beyond an overnight fast, say 1-2 days, glycogen runs up pretty quickly then, right?
It does. Liver glycogen is mostly gone within about 24 hours. So the body has to compensate by dramatically increasing gluconeogenesis. This is driven by falling insulin and slightly rising glucagon. Muscle protein breakdown accelerates. More amino acids like alanine are shipped to the liver, which ramps up its glucose production using them. Clinically, you see this as increased nitrogen excretion in urine. You're losing lean body mass.
That sounds costly.
It is. And it's not sustainable long term. Simultaneously, lipolysis really cranks up. Hormone-sensitive lipase is highly active. Plasma fatty acids rise, providing fuel for muscles and other organs, further sparing glucose for the brain. Interestingly, high fatty acids can actually cause a bit of insulin resistance in muscles. This helps spare glucose during fasting, but it's also implicated in the problem seen in obesity and type 2 diabetes. These fatty acids flood the liver, providing energy for gluconeogenesis. But if they arrive faster than the citric acid cycle can handle them, acetyl-CoA builds up and ketogenesis begins in earnest. Ketone levels start to rise in the blood. Now, for prolonged starvation going beyond a couple of days, there's another major metabolic shift. The body switches priorities again to preserve protein at almost all costs. How? By relying heavily on fat stores and ketones, and crucially, by teaching the brain to use ketones as its primary fuel. So protein breakdown dramatically decreases. Urea excretion plummets. This is key for long-term survival. Liver gluconeogenesis slows down, partly because less alanine is coming from the now protected muscle. But the kidneys increase their gluconeogenesis, compensating significantly, maybe providing up to 40% of glucose production. This might be linked to handling the acid load from ketones. Lipolysis and ketogenesis are maximal. Plasma fatty acids stay high. The liver churns out ketones, maybe 100 grams a day after a few days. Low insulin also means peripheral tissues don't grab ketones as eagerly, so blood ketone levels soar. This forces the brain to adapt. It increases its uptake and use of ketones, eventually getting more than half its energy from them.
So the brain learns to run on fat indirectly.
Essentially, yes.
Hmm.
This spares glucose, which means less need for gluconeogenesis, which means less need to break down precious protein. It's an incredible adaptation. There are other hormonal changes too, like falling leptin levels, which can affect reproductive function, a protective mechanism during famine. Ultimately, survival depends on fat stores. When they run out, the body is forced to break down essential proteins, leading to muscle wasting, organ failure, and often infection. But the brain itself is usually kept fueled until the very end.
Wow. Okay, so summing this all up, we've really covered a lot of ground. From the definition of metabolism, through the different fuels, the energy laws, the core pathways like glycolysis and gluconeogenesis, fat handling, and these incredible adaptations to feasting and fasting.
It really does showcase the body's incredible intelligence, doesn't it? And its resilience. Understanding these fundamentals, glycolysis, gluconeogenesis, beta oxidation, ketogenesis, it's not just about memorizing pathways. It's about seeing the logic, how it all fits together, and how disruptions lead to disease.
Absolutely. And that's your deep dive shortcut to getting a handle on metabolism. The human body is just amazing in its adaptability, constantly striving for that balance. So maybe here's something to think about. We've seen how remarkably flexible the body is in switching fuels. How could we maybe leverage that natural metabolic flexibility, that ability to shift between carbs, fats, ketones, maybe in new ways to optimize health or even treat metabolic diseases?
That's a great question to ponder. And remember, as you're wrestling with these concepts, you're building a really strong foundation. Keep asking questions. Keep digging deeper. Know that you're part of the Last Minute Lecture family, and you absolutely have what it takes to master this stuff. We're definitely here to help you along the way.